probably always interested in the idea of being a scientist. It came largely from reading books. Aerosmith was one of the books that influenced me a great deal. Microbe Hunters was another. It sort of glorified the research ethic, and for that reason, I guess I always dreamed of wanting to be a scientist. And I was fortunate when I was in high school that there was a woman who loved young people and organized an after-school science club. And I participated in that club, and the remarkable thing about her, she was not a teacher. She was the woman who managed the stockroom and made sure that all the right equipment was out for the physics class and the chemistry class and biology classes. But in the setting of the science club, she was remarkable in the sense of always getting you to find the answers to the questions you asked. So whenever you raised an issue or a question, she would frequently goad you into trying to find the answer yourself, either by going to the literature or in fact doing an experiment. And so my first experiments were actually done in the context of a high school science club, which certainly was a very stimulating setting. As an aside, the interesting thing about this high school teacher is that she also influenced Paul Berg and Jerry Carl, all three of whom won the Nobel Prize. And so some 10 or 15 years ago, the city of New York named one wing of the high school as the Sophie Wolf Science Wing, and one floor is named for me, and one floor is named for Arthur Kornberg, and one floor for Jerome Carl. So she obviously had a big impact in stimulating people at that stage. When I graduated from high school, it was during the war, it was 1943, and I had decided that at that time that probably being an engineer was more important than what I had my medical research idea. And I went to City College in New York. I lasted 3 days because it took me about 2 hours and 3/4 to get to City College from where I lived in Brooklyn, and 2 3/4 hours back. And so after 3 days of this kind of travel, I decided to chuck it. I didn't want to be an engineer. And so I went to attend Brooklyn College, which was much closer to home. And it was more or less marking time. And so I spent 1 semester at Brooklyn College. And at that time, I realized that biology wasn't what I was really interested in. Biology as it was being taught at that time. A friend of mine who had gone to Penn State in the engineering school sent me the catalog. And when I thumbed through the catalog, I saw that there actually was a department of biochemistry. I'd never heard of biochemistry before. It sounded like it was not biology and not chemistry and just what I might be interested in. So I went to Penn State to study biochemistry. Shortly after I got there, I entered the Navy, and the Navy sent me back to Penn State to continue in studying biochemistry, but also taking a series of courses that would prepare me for going on board a ship. I did quite well in biochemistry, and during my last year, when I came out of the Navy, I went back to Penn State to finish up, and during my last year, I did a series of papers on the newly emerging isotope technology, radioisotopes, and I read a lot of the papers in the literature, and I was astonished at the ability to be able to follow metabolic reactions by using radioactive tracers. Most of the papers I read came from a place called Western Reserve University, which I'd never heard of before, and they were largely coming from a laboratory of a man named Harland Wood. So I decided that I wanted to get into that area, and I applied, and I was accepted to go to Western Reserve, but when I got there, I found out that I was actually in the wrong department. I had applied for and been accepted to a department called clinical biochemistry, and the biochemistry department was newly created to largely supplant this old-fashioned clinical biochemistry. And so for 2 years, I actually worked in clinical biochemistry, developing the artificial kidney and learning to do surgery and removing kidneys from dogs and keeping them alive, and also trying to isolate rare proteins from urine using the kidney as a dialyzer to get rid of salts and material like that. Eventually, after 2 years, I was taking courses in the biochemistry department. Harlan Wood came to me and asked me if I would like to transfer into the biochemistry department, which I did. And for the last 2 years of that 4-year period, I did much of the work that used tracers and studied C1 metabolism and the synthesis of methionine from formate and formaldehyde, and actually was the first to discover the role of folic acid and B12 using in vitro system for the synthesis of methionine. During the last year of my graduate work, I began to think about what I wanted to do next. Harlan Wood had in mind for me to go to work with Paul Berg, who at that time was the preeminent biochemist in the world, pretty much. He was doing it largely. He thought that was the right step for moving me up the scale in my career. But I had already made arrangements to go to Copenhagen to work with Herman Kalkar, and for a second year to come to work with Arthur Kornberg at the NIH. So Harlan Wood was flabbergasted of why I would not go to St. Louis. I was embarrassed to tell him, but eventually did, that I didn't want to live in St. Louis because the weather there was abominable. And I'm sure that in his estimation, my standing went down several notches, that I would sacrifice going to work with Paul Berg for my distaste of living in St. Louis. As it turned out, we had a great year in Copenhagen. It was very productive. It was culturally a terrifically exciting year. And mid-year, I received a letter from Arthur Kornberg saying he had decided to move to St. Louis, to Washington University, and would I still like to come? I ultimately decided that working with Kornberg was at least enough to get me through a year of bad weather in St. Louis. As he told me later, out of 6 or 7 postdocs that had agreed to come to his lab, when he put the same question to them, all but me, I was the only one who agreed to go to St. Louis. All the rest dropped out. I went to St. Louis as a postdoctoral fellow, and after the first year, the work was going very well, and Kornberg asked me to stay on the faculty. I actually received a scholar appointment from the Cancer Society, and so for 2 or 3 years, I was sort of a high-level postdoc, if you will. And as soon as the opening created in the department, Kornberg appointed me as assistant professor. This was a microbiology department, and Kornberg recruited some really amazing people: Dave Hognes, Dale Kaiser, Mel Cohn, and it was one of the probably the most exciting time for me in science. This close-knit group, we were all about the same age. We didn't know each other beforehand, and we worked in completely different fields, but the mechanism that Kornberg had set up of maximum exchange between people. We ate lunch together every day. We had a noon seminar during lunch every day, and we were able to essentially educate each other about our own fields to the point where you actually could say to somebody, that experiment worked, and that person knew what experiment you were talking about. After about 5 or 6 years, the department was doing extremely well. Kornberg was invited to come to Stanford to be the director of the first biochemistry department. Now, Stanford had never had a biochemistry department as part of the medical school. Biochemistry was taught to undergraduates who were here for the first 2 years before they went up to do their clinical work in the city. When the trustees decided to move the medical school down to Palo Alto and put it on the campus, Henry Kaplan, who was really one of the powerhouses in the medical center, strongly urged that they go and recruit Arthur Kornberg, who at that time I think was largely considered as one of the leading young biochemists in the world. He assured me he was not interested in moving from St. Louis because the department was growing up very well. But one visit to the campus was sufficient for him to come back and said it would really be a great, great opportunity since we would be able to move into a totally new laboratory designed by us. But he also said he would not move, accept the job, unless we all agreed to join him. Well, after about a month or a couple of weeks of discussing the move and generating a lot of excitement about that possibility, we all agreed that we would move to Stanford to create the new biochemistry department. This was 1957, 2 years before we actually moved. So for 2 years we were lame ducks, and that wasn't the most pleasant thing, but it gave us sufficient time to actually plan a totally new kind of biochemistry course and to design the physical facilities and all the labs and to accumulate all the things we needed to take us. So when we moved in June of 1959, we moved into the building after a couple of weeks of sort of hanging around, but we were all doing experiments by mid-August. Now, people look back at the biochemistry department and marvel. It had several unique aspects to it. One was an extraordinarily close-knit and friendly group. And in the 50 subsequent years, there's never been any bad feeling amongst this original group. Of the 7 who came, only Melvin Cohn is not here today. He's at the Salk Institute. So 6 of the 7 have remained here through 46 years, and to this day, still remain close friends and colleagues. One of the unique features of that department was the culture that Arthur Kornberg established. I mean, just to give you an example, when I joined him as a postdoc in St. Louis, shortly after I became a faculty member, he told me that I need not apply for a grant right away, that I could certainly use his funds to do whatever I needed. And so for about the first year or two that I was on my own, I didn't have an NIH grant. I was actually working using funding that he had. And then he urged me to apply for a grant, more not because I needed money, but because he wanted me to get into the process of the grant review system. When we came here, pretty much that culture pervaded everything. We were together as a family, we shared everything. So one of the unique features that people remarked on was that nobody had assigned space. Students and postdocs were placed wherever there was an empty bench. We each had a lab and an office, and we had 1 or 2 people in our own lab, but the rest of our groups were scattered around the department. And one of the extraordinary features about that was that each of our students learned a great deal more than what was just going on in our own group. And frequently, our group meetings were peppered with somebody saying, "Did you hear what happened in Kornberg's lab?" Or, "Gee, Dale Kaiser developed this neat trick." And so we had this feedback thing. There was enormous sharing of equipment, sharing of supplies. I've said this many times that almost all the reagents that were used in the first experiment to make recombinant DNA were in Kornberg's refrigerator and were made available to me freely for use for the work we wanted to do. And in addition, up until the NIH eventually put a lid on it, we shared all our funds. And since our field, what we were working on was somewhat related, you could easily justify an expenditure if it was going to be used by somebody else. And so for many years, we essentially had a departmental fund from which we all drew. And that was extraordinarily beneficial for young faculty, because young faculty, when they came in, were not limited to what they themselves could raise in funding. They were only limited by their ideas and their ability to get things done. And I think some of our young faculty prospered enormously early on, even though they couldn't have brought that amount of funding to their program. I came to Stanford as a biochemist. I was mostly interested in enzymology and had been spending much of our effort on understanding the way amino acyl adenylates were used to form the amino acyl tRNAs. And much of the early work was involved in protein synthesis. So in a way, I'd made the transition from what was a classical biochemist to what people began to call molecular biologist. And so I learned a lot of genetics. I learned about bacteriophage that I never heard of before. And I guess I could have easily claimed to have been a bona fide molecular biologist. And of course, I was well aware of the remarkable work that was coming out of the Pasteur Institute, elucidating the mechanisms of gene regulation, repressors and operators and things of that sort. And one day, I think I sat down and said, is all this that happens in bacteria the way it works in higher organisms? And I must admit that I was taken by the fact that the focus was primarily on E. coli and that system, and very little was being done about mammalian systems. And in fact, what was being done was very confusing. And so I was sitting in a lecture by Dale Kaiser, who was giving a course on lysogeny, or on lambda-phage, and the last lecture was on lysogeny, and he was describing how the lambda-phage integrates into the chromosome of the infected cell, and then changes the character of the cell by the genes that the lambda-phage expresses or influences. At the end of that lecture, he described the analogous system which had just recently been discovered of tumor viruses. Animal viruses, which infect mammalian cells, integrate into the chromosome and express one or a few of their genes and transform a normal cell into a tumor cell. And I was really turned on by that observation because, having recognized the important role that viruses had played in elucidating many of the mechanisms of gene function and gene regulation, it seemed reasonable that beginning to study a small animal virus might give us a handle on learning something about the mechanisms that exist in mammalian cells. So I decided to take a year's leave of absence. It was the only sabbatical I ever had, really. And I went down to the Salk Institute to work with Renato Del Becco. Renato Del Becco was the discoverer of polyoma as a tumor virus, and some years later, SV40 was discovered to also be a tumor virus. And so I went down there to actually learn how to work with animal cells in culture and to use the polyoma as a way of trying to analyze gene function in mammalian cells. Interestingly enough, when Renato was asked if I could come, he embraced the idea, told me I could come and be welcomed. I had already agreed to have a fellow from the Pasteur Institute, Francois Cousin, come and join me as a postdoc here. And so, in fact, I, Francois Cousin, and my longtime assistant, Marianne Diekman, all went down to the Salk Institute and became experienced, if you will, in the animal culture. Now, one of the things that was really very impressive is before I'd done that, I'd worked with Charlie Yanofsky, and Charlie had educated me to the power of genetics and the ability to be able to move genes from one cell to another using phage-mediated transduction. And I became pretty facile in thinking about that technology and using it, and it was one of the great experiences of my career was collaborating with Charlie on one or several papers. But that experience convinced me that what we needed to do was to develop a comparable system for being able to introduce new genetic information into mammalian cells. The ability to do that in bacteria had facilitated the design of experiments and what you could actually attempt to do. And so I thought it would be important to see if we could use the tumor viruses as a mechanism for transferring DNA from one cell to another. As it turns out, the tumor viruses have very small genomes. There's only about 5,000 base pairs. And so there's not a lot of room to introduce foreign DNA into it and still have a virus particle. So when we realized that the virus itself could not be a carrier, of genes from one cell to another, it became obvious as could we devise another way to do that, and the idea was to use the viral DNA itself, which was known to be infectious, known to integrate into the chromosomes of cells it infects, and so the idea was, could we actually insert a new piece of DNA into the viral chromosome in vitro, and then transfect cells with that new hybrid DNA and get those genes into the chromosomes of the infected cell. And that was the idea of creating this recombinant DNA. We didn't use the word recombinant DNA at the time. It turned out to be straightforward because we knew and were aware of that lambda phage uses what we call cohesive ends. And cohesive ends allow two DNAs to join each other. Now, SV40 doesn't have cohesive ends. At least we didn't know that it had any. And so we cut the SV40 DNA circle open and synthesized synthetic tails onto the ends of the DNA, and then took a piece of lambda phage DNA and put complementary sticky ends onto that, and then just by mixing the two, they joined together to form a SV40 lambda phage DNA recombinant. The molecule we made, which contained all the genes of SV40, including the genes which were known to induce tumors, was joined to a bacterial piece of DNA, which itself had the capacity to replicate in E. coli. So this two-headed molecule had the potential for being able to be replicated in E. coli and also to integrate into the chromosome of mammalian cells. It was our intent to be able to do both and to study whether bacterial cells could read mammalian virus genes and whether mammalian cells could read the bacterial genes. One of my students, Janet Mertz, went to Cold Spring Harbor to take a summer course in tissue culture, and in the course of that visit, she described what we were doing. And a man named Pollock, who was teaching the course, was outraged by what we were attempting to do, and he called me to express, saying it was the most outrageous, stupidest experiment he could possibly imagine, because here we stood on the frontier of possibly spreading tumor genes throughout the population, because E. coli inhabits our intestinal tract, and the concern was that this recombinant DNA could enter into a person's intestinal tract via E. coli and cause cancer. I thought the whole argument was bunk, and we argued back and forth. He began to speak more broadly about this impending danger, and I became more and more defensive. Ultimately, I spoke to a lot of people whose opinions I respected, and I think I came to the conclusion that I could not, with 100% assurance, say that the bacteria carrying this recombinant DNA could not escape from the laboratory and infect people. I thought it was highly unlikely. I thought even if it occurred, the likelihood that that DNA could get out into the intestinal tract and infect the cells there, but I was not 100% sure. And so we decided to put the experiments that we were going to do with the recombinant on hold. We're going to try and address the issue of whether each of these mammalian cells and bacterial cells could express foreign genes, tested another way. In the meantime, one of my students discovered that EcoR1, the enzyme we had used to cut ASV40 DNA, actually made cohesive ends, something we had not known. And so we had synthesized these cohesive ends of A's and T's, but now it became quite obvious that naturally occurring cohesive ends could be used to join any two DNA molecules together. Now, Stanley Cohn, who was working in the biochemistry department, although he was in the Department of Medicine, was well aware of these experiments, and actually he and Herb Boyer, who was the discoverer of EcoR1, and who also was made aware that the enzyme made cohesive ends, joined together and began to do experiments of actually joining two different kind of bacterial plasmids together. Actually, one of my students, John Morrow, had the idea that maybe one could actually insert into a bacterial plasmid some eukaryotic DNA. Now, he was targeted to go to Don Brown's lab to work on ribosomal DNA. And he had some ribosomal DNA in his refrigerator. And so he cut that with R1 and joined it with the plasmids in Stan Cohn's lab that had R1 and created the first bacterial eukaryotic DNA, which they were then able to introduce in bacteria and clone. And that was a very, very critical step, the actual amplification of a recombinant DNA in a bacterium. Once it became clear that you could join any two DNAs together and replicate them in bacteria, there were any number of what ifs. What if somebody puts in a toxin gene into E. coli? What if somebody puts in a drug resistance gene? Could this be spread around? And so very early on, there began to be this gathering concern, if you will, about the safety of this whole technology. And the first warning or message came out of a Gordon conference in which the members of the conference heard Stan Cohn and Herb Boyer present their data and then held a rump session at the end of the Gordon conference to agree to notify the president of the National Academy of Sciences that, in fact, this research had enormous promise, but had certain potential risks. And they did draft a letter that was published in Science. It was sent to the president of the Academy. And the president of the Academy received it and said, well, what do I do with that? And Maxine Singer, who happened to be the organizer of that Gordon conference, told them that he should contact me because I had already worried through this issue. He did. I was in the midst of teaching a course, and so I deferred doing anything. But when the course was over, I organized a small group, Dave Baltimore, Jim Watson, Dan Nathans, a bunch of the people who were likely to be able to understand and use this technology. We met at MIT and we drafted what came to be known as the moratorium letter. It wasn't really calling for a moratorium. It identified 3 experiments that we told people to defer. One was not to introduce toxin genes, the other was not to introduce cancer genes, and the other one was not to introduce drug resistance into organisms that are infectious. But all other experiments were left open. But the world interpreted it as us calling for a halt in recombinant DNA. And in Britain, of course, they did have a complete halt. In this country, cloning E. coli genes in E. coli seemed to be innocuous, and that was going on. The letter called for holding an international meeting 7 months hence, and that was the Asilomar Conference. And the Asilomar Conference was an attempt to bring together people who were experts in infectious disease and in various areas, containment, for example, that might allow this research to go forward. That conference was held, and after 3 days of rather contentious arguments, all of which are detailed in picturesque books, the conference decided to recommend that the so-called moratorium be lifted, but that strict guidelines be developed to inform potential researchers in this field. And that's the origin of the NIH guidelines, which in fact proved to be extremely helpful and useful because most molecular biologists were totally unaware of the problems of infectious disease. Nobody had ever worked with pathogens, or very few. And although these turned out not to be pathogens, the concern then was that they should be treated as if they were pathogens. And we knew biochemists didn't even know which end of a loop to flame. So the guidelines were an educational tool as well as satisfying the public's concern. And I think the important element about the Asilomar Conference was it generated, in my belief, trust of the scientific community to be straightforward and honest. And all of the meetings that had to be held here at Stanford in regarding any of this experiment that was open to the public. And for the first few months, the public did attend. Eventually they saw that we were not trying to fudge anything, and they didn't understand what we were talking about anyway, and so the public eased out, and the research progressed. And I think I'd say, I believe, at least, I don't think any of us at that time foresaw the full implications and the blossoming of what recombinant DNA would do. I think we could see what we call the low-hanging fruit. You could see the first thing, making growth hormone, making insulin, things of that sort. But by and large, the full development of what that has accomplished, I think none of us foresaw. And you can look back now with pride and say, I think we did the prudent thing at the time. Jim Watson, I know, has a very different view. He thinks it was the stupidest thing he ever signed on to. And even to this day, he thinks we impeded science. But in fact, rather than impede science, I think we helped it move along faster just because we generated the confidence and the trust in the public that we were doing the right thing. You know, that's a favorite, it's a favorite question of people to ask, how has the Nobel Prize changed your life? I think it's probably changed my wife's life more than mine, because I think she was thrust into a kind of limelight that she doesn't particularly enjoy. And as you can tell, I certainly don't shrink from the limelight, so I've been involved. I think the thing that has disturbed me the most about the prize is the expectation that people have of you of having the prize. So you're accorded the privilege of ultimate wisdom, particularly on topics that you know nothing about. And also, people love to be associated with a Nobel Prize winner. So one anecdote that illustrates that thing is when we were raising funds for the Beckman Center here, I had to go attend a dinner down in Los Angeles, and the woman sitting next to me was the wife of a potential donor. And so we entered into a conversation around topics which were really fascinating. Her husband was involved in the movie business. My son at that time was in acting school. We both collected art. And so we had lots of topics to talk about. At the end of the dinner, we parted company, saying how much we enjoyed meeting each other and the conversation we had and so on. A few minutes later, she came rushing up to me and said, "Young man," waving her finger in my face, "I am really very angry with you." And I was taken aback because I couldn't understand why she would be angry. She says, "Never once did you mention that you are a Nobel Prize winner." And that's a kind of reaction which, I was at a press conference yesterday. I was introduced to the governor. Paul Berg is a Nobel Prize winner. To Senator Feinstein. Paul Berg is a Nobel Prize winner. As if that is supposed to give me a very special status. Even here at Stanford, frankly, Stanford loves to parade what I call running up the Nobel flag on its flagpole. And we have tremendous people here at Stanford, extraordinarily talented, able, wiser than I am in many areas, but they always seem to want to call on a Nobel Prize winner to make the presentation to the incoming class or to this group or so. So that's the part of the whole thing which I think I have done. On the other hand, there's no question that it opens doors and has given me access to people and occasions and places that I probably never would have been allowed in before. So there's that balance. It can be overused. It does serve its purposes. Lots of petitions with Nobel Prize winners. They often sign their names onto things they have no idea about. Nevertheless, it has cachet. And so that's the plus and minuses. The other thing is being invited annually to the Nobel dinner and the affair. It's a great event. And we enjoy doing it whenever we're invited. One of the things that was most important when we came to Stanford was that we were all going to be teaching biochemistry, which none of us had done before. Dale Kaiser was a geneticist. Dave Hogness was more a kineticist. Arthur Kornberg was an enzymologist. I was a metabolic enzymologist. But we all had to teach biochemistry. And so we all had to learn every aspect of the course so that each of us could teach it in a moment's notice. We all sat in on each other's classes. And at the end of each lecture, we would often discuss the good points, some of the bad points, how you might better have used the blackboards than you had you used it. So we developed a strong ethic that teaching was very, very important, and that the medical students were fun. And our laboratory course in that thing, which was the highlight of the biochemistry course, was essentially devising experiments on the spot. We knew we could bring any reagent down that would enable any student to do any experiment they suggested within certain boundaries. Half the course was enzymology, half was DNA. There were papers published out of the experiments that the medical students did. We had complaints from other departments that the medical students were spending far too much time in biochemistry laboratory and not attending their courses. So teaching was a very important part. I think I grew up, all of us took pride in the quality of the course. It was always rated the best course in the medical school, and so on. A lot of things changed in the late 60s during the Vietnam War. And suddenly the students were much more motivated about politics. They were motivated about going out into the valley to work with the poor farmers than doing DNA biochemistry. And so the course began to sort of fade a little bit. Then, of course, there was all of the ruckus going on campus about anti-war stuff. And I remember when the U.S. invaded Cambodia, I was one of the faculty that was chosen to go to Washington to speak to the Congress people about what was happening in Vietnam and Southeast Asia. There were teach-ins, there were sit-ins, there were protests, and we had a particularly serious event happening in the medical center, where a group of people broke in and barricaded themselves in the blood center and refused to come out unless their non-negotiable demands were met. And after about 3 days, the police came in, banged a lot of heads, destroyed a lot of the books. And so the mood on campus was the blackest I can ever remember. One of those afternoons, I had a visit from 2 of the medical students who came to me with the idea of making a movie on protein synthesis. Now, I'd been lecturing on protein synthesis at the time, and they prided me in saying that I'd made the process seem so dynamic that they thought they could actually illustrate the whole process in dance. Each of them had a girlfriend who was a dancer. Each of them had had filmmaking experience at UCSD. And so they came to me to ask me, I was chairman of the Department of Biochemistry at the time, could I provide some funding for them to make this movie? I thought they were pulling my leg. I couldn't believe, with the mood that there was on campus, that somebody could come with such a flip idea. And so I was absolutely delighted to provide them with funding, hoping it would change the mood and things like that. And so they got me involved in the making of this movie. And the movie was to use people playing the role of molecules. And so there were going to be 30 people for the 30S ribosome and 50 people for the 50S ribosome. There would be a messenger RNA of 54 bases. The bases were coated by balloons, colored balloons attached to the head, blue, yellow, red, and green for the 4 bases. And the film was going to be made with a camera mounted on the top of the 8-floor high dorms. And it would be carried out on the grassy green below the dorm. And they pulled this off in a magnificent way. It was, I remember thinking as I went to watch them rehearsing, it was like football practice. They had the 30S ribosome with one guy with a clipboard giving the people instructions, another one for the messenger RNA, the various initiation factors, and so on and so forth. And they asked me to do the introductory bit. And I did this in dead seriousness because the film had not yet been shot at the time I did my introduction. But it was to explain protein synthesis and how it was going to be portrayed in this dance idiom. The film was made, I wasn't there at the time. The students took the raw film down to them to San Diego. They edited and made a 13-minute piece out of it and put in the rock music and dubbed in all of the Jabberwocky sounds. And they showed it here for the first time at the Stanford Flicks. I was away out of town, but my wife and son went to see it. And it was the most raucous event that people could remember at that thing. It really went over very big. It is used widely. It was reviewed in Nature, saying that it was accurate. It is accurate to this day, now 30 years later. It was filmed in 1971. And it is accurate to this day. There's a little more subtlety in some of the details, but my contribution to the film was how to portray the use of high-energy GTP for the coupling step. And I suggested that we get a small fire extinguisher and be able to put some flour into the scoop, the funnel, and every time the person who was wearing a kind of a black cape would run into the place where the hands were being joined to signify the creation of a covalent bond, he would pull the trigger, and it was a big puff of flour of smoke, so to speak. And if anybody sees the film, most people remark that that was absolutely one of the highlights of it. So my contribution was the 3-minute introduction, which was deadpanned, and the GTP smoke. Anyway, it was a great film. The students loved doing it. I don't think we had to, in order to get enough people, we had to recruit people from the street. So there were little kids, and everybody was wearing hospital gowns. The musicians who played the rock music that accompanied the thing were all dressed in the attire that was characteristic of the 70s, bushy hair, long hair, torn jeans, you name it. But I still get requests for the film. We have made it into DVDs, so it's available. We just send out a DVD to anybody that asks. People have seen it multiple times. So, for example, when we were teaching medical students, they had seen it in middle school, they'd seen it in high school, and they'd seen it in undergraduate biology courses, and they still wanted to see it in the medical school biochemistry course. Since I retired as professor of biochemistry, I've become heavily involved in public policy issues. I suppose I had my baptism of fire during the recombinant DNA controversy when I did a lot of speaking in Congress and defending recombinant DNA and helping develop the guidelines. When the stem cell issue broke in 1997, not being a stem cell biologist, but recognizing that this was an important breakthrough, an exciting thing. It had the same qualities as the recombinant DNA, sort of a sudden breakthrough, broadcasting lots of opportunities, and in contrast, and similar to the recombinant DNA, the initial kickback and the concerns about whether we should move in this direction. I suppose I was probably more outraged by the way Congress responded to the stem cell breakthrough, by essentially heavy-handed proposals to essentially not only block any stem cell research in this country, but also to actually prohibit access to any therapies that might be developed using that technology. And that in itself was an arrogance which just turned me on. So I became very much a spokesman and did a lot of testifying in Congress, public groups, and so on. Proposition 71 emerged from actually the California legislature having passed two bills, one which prohibited any attempts to clone people, and the second authorized stem cell research, including what people call the therapeutic cloning, but which we now refer to as somatic cell nuclear transfer, authorizing it to be done, but there was no funding actually appropriated for it. So it became clear that with the state not only being eager to support this line of research and being out front, we had to find a way to get the money. And since there was no prohibition against use of private funding, the notion emerged that perhaps we could begin to go to the citizens of California and ask them if they would support this research program, which had the promise of developing cures, new knowledge, and so on. And Robert Klein, who's a local realtor, was very experienced in the idea of bond issues. He had actually pioneered the use of bond issues for low-income housing in California. And so he drafted this proposition to go on the ballot as a constitutional amendment, which would authorize the creation of $3 billion worth of California bonds to be issued over a period of 10 years to support human embryonic stem cell research in California for a period of 10 to 15 years. That was a breath of fresh air. It provided a way to get around the president's prohibition on the development of new stem cell lines and the threat that they're going to make criminals of people who wanted to proceed with certain lines of stem cell research. And so I became quite active in supporting the Prop 71. I spoke frequently at various citizens meetings around the state, met with legislators, and it passed. It passed with a majority of more than 59% of Californians voted. 7 million Californians voted to support what others identify as cloning, but which is in fact the creation of what I call patient-specific stem cells for therapeutic purposes. So it passed, but I think like any enterprise of that size, starting out from scratch, the NIH has had 50 years of experience of working out mechanisms for giving away money. This initiative will be giving out $300 million a year in research support, grants, facilities, teaching, educating people. Really, the whole idea is to build up a huge enterprise that would promote stem cell research for both basic and therapeutic goals. One of the things to emphasize about the implementation of Prop 71, and that is the opposition is trying to find ways to challenge its legitimacy by challenging whether it's bypassing the normal legislative process of apportioning money. But in fact, all of that was spelled out in the proposition. That's what people voted for. So all of those challenges will disappear because the state Supreme Court will certainly rule them as being moot. But nevertheless, it is a huge undertaking to try to put in place a process of giving away $300 million a year and doing it in a way which is open, legitimate, without conflicts, and will ultimately achieve the goal of trying to create the new knowledge upon which medical advances will rest. And so I have been strongly supportive, and when I was in Los Angeles yesterday, it was to help support Senator Feinstein's bill pending, which is to outlaw human cloning without the attachments of trying to impede the research needed to develop the therapies. And it's a bill which I regarded as nearly scandalous that the American Congress, after 8 years of being alerted to the fact that people's concern about attempts to clone people, has still not passed any legislation. California has it in its constitution, but none of the other Congress has. So I think I said yesterday, California is my adopted home of the last 46 years. And I could never have been more proud of this state by the way it responded to the Prop 7 initiative and the way it's responding today in making it a reality. The JBC was the premier journal for publishing advances in biochemistry. There wasn't much of anything else. There was the European Journal of Biochemistry. I don't know if I ever published anything or even considered publishing anything there. When I was a postdoc in Denmark, the work I did there was published in the JBC. When I came back and started my work with Paul Berg, sort of the major discovery of that stage was published in JBC in a series of papers. And I think I never thought of any other journal for a very long time. I think it was only when my interest shifted to the work that led to the recombinant DNA that I began to publish stuff in other journals. And that was because it wasn't appropriate for JBC. But JBC has certainly been an extraordinarily influential journal, and its standards are unrivaled by anybody. I'm always amazed to see how the editorial board has grown in number. It used to be relatively small. I think I served at least as a referee on many JBC papers. And so its standing remains as high as it ever was. One of the things that I benefited from was Kornberg was very, very helpful as an editor. So at that time, again, it was a part of the culture of the department. We exchanged each other's papers before they were submitted. So we got a lot of feedback from colleagues. Kornberg had this red pen that he used very judiciously, and he helped me to learn how to write a paper. And so I don't remember that I had any problem with any of the papers I submitted during this early part of my career. In large part, they'd been already worked over by very critical people. And in terms of the styling and the content and arguments. So that was a good learning process, and it benefited me enormously. I think one of the things that I learned during all the time is that science is an activity which is filled more with failures than successes. Not every experiment gets published. You have notebooks and notebooks full of experiments that failed. And a quality which I regarded as imperative, because to me it was something always in front of me, is the resiliency of coming back after having worked all night and gotten nothing, or a result that didn't make sense. And the ability to come back and reformulate and redesign the experiment, rethink the experiments, rethink the data, and so on, was to me the most important quality. Because I've seen in other people give up too easily. And if things don't work right away, they go on to other things. But I think most of the important work I think that I've accomplished has been building on negative results and eventually succeeding. So that's an important one. The other thing is, I think, something which today is maybe even harder to do. The field is so active, it's hard to think of something to do that isn't being done somewhere. And so it's probably discouraging to young people because, in a way, they come out of their graduate work or postdoc and almost anything that they want to do is being done. And so it is critical for being able to think outside the box. And I think I was encouraged always to think big, aim high, somebody would say. Don't do what everybody else is doing. Try and find new ways to answer questions. I've told my students all along that if you have a crazy idea, do the experiment to test that crazy idea, because you're almost surely going to find something new. Because if nobody has ever done that crazy experiment before, you're likely to discover something new if you have your eyes open. And so I think I encourage crazy ideas. I used to bet my students on the outcome of the experiments. I'd almost always take the side that was almost surely going to fail, just to encourage them to think that they might succeed and win the bet. And students have won many bets because they did ultimately find something that was totally unexpected. And I think that's something to encourage people to do, is to follow crazy ideas.